Welcome to the BioWhisperer channel. Our topic today is on a perspective overview of the clinical metagenomics. With the popularity of next-generation sequencing tools and its widespread use, researchers are moving towards the deployment of the next-generation sequencing of pure culture isolates in clinical microbiology laboratories has the potential to revolutionize the fields of food safety and public health. Metagenomics and other omics disciplines could provide the solution to a cultureless future in clinical microbiology for the identification of clinical causative agents or foodborne contamination, detection of AMR and/or virulence factors, in addition to providing high-resolution subtyping data. In areas of food safety and public health, metagenomics assays may provide a universal test for clinical diagnostics, foodborne pathogen detection, subtyping, and investigation. As for the utility of metagenomics in a diagnostic setting, researchers noted, as is the case with many infections, differential diagnoses can be extensive. Consequently, cultures, serology, and pathogen-specific PCR can be difficult in determining etiological causation. Due to the need for reference databases such as UniProt or Silva being cornerstone of NGS-based diagnostics, level of curation varies across platforms. Often, there is an additional developmental and modernization need for analysis tools and bioinformatics pipelines. This involves expertise to help with the computational and technical challenges of analyzing data accurately and in a clinically actionable timeframe. Apart from that, sample preparation and sequencing methodologies must also be standardized. Other factors that can affect pathogen identification include, as previously discussed, pre-enrichment steps, quality of DNA extraction, library preparation, and chemistry. Furthermore, we are faced with questions pertaining to how to work with contaminants or host DNA, low biomass specimens, defining ideal read depths for particular biological specimens or food products, and lastly, defining sequence number thresholds to confidently assign pathogenic etiology. Next section, we have a quick discussion of metagenomics as an unbiased methodology. Viruses are much smaller than bacteria, and when we perform wet lab preparation, the amount of viral genomic content represents a microscopic fraction of total DNA or RNA in a given sample. Care and caution need to be interrogated with high sensitivity within the protocols of a metagenomics assay. Though shotgun metagenomics can produce near-complete whole genome coverage of highly abundant microbes, achieving sufficient sensitivity in shotgun metagenomics studies has proven to be a challenge, particularly with respect to low-abundance microbes and gene-level identification. Metagenomics sequencing of a clinical or environmental sample could offer a universal test for pathogen detection, clinical diagnosis, as well as a subtyping test for routine surveillance activities. By tracking foodborne pathogens along the farm-to-fork to clinical specimen continuum, it is possible to monitor trends over time, including factors or issues implicated in causing illnesses and detecting outbreaks. Recent improvements of sequencing technologies and streamlined bioinformatics tools have not only advanced clinical diagnostics but are also transforming public health. Effective detection of outbreaks, particularly broadly disseminated outbreaks caused by the commercial distribution of contaminated foods, is largely dependent on subtyping isolates from a sizable proportion of cases. The use of shotgun metagenomic sequencing to identify antimicrobial resistance genes is promising, and studies are now beginning to elucidate their plausible role in surveillance. Early work with metagenomics reads are mapped against a database containing a comprehensive catalog of known AMR genes have variably identified a large abundance of AMR genes were detected in the human gastrointestinal tract. Most studies applied shotgun metagenomics sequencing to detect AMR genes. The optimization of the workflow involved determining microbial abundance reference ranges. From a diagnostics perspective, limits of detection need to be established in order to have sufficient coverage with the capacity to detect rare AMR genes or in complex metagenome. Functional metagenomics, on the other hand, is an exceedingly powerful technique attributed to its capacity to discover novel and highly divergent AMR genes. Functional genomics involve cloning total community genomic DNA into an expression vector and transformation into a susceptible expression host where the transformant library is then assayed for AMR genes are sequenced and annotated. Irrespective of how and when NGS technologies will be capable of rapidly and accurately detecting AMR and associated susceptibilities, difficulties in public health AMR surveillance will be apparent. As for the perspectives for the future, researchers realize that the preceding widespread implementation of metagenomics sequencing and analysis in diagnostic and public health settings will require an accompanying ecosystem of advancement in bioinformatics encompassing algorithms, software and pipelines which will need to be developed, validated and standardized through various ring trials and retrospective analyses. Creation of a global database that houses uniquely identifiable microbial genomic data in combination with high-quality meta and epidemiological data. Lastly, we feel that public health surveillance will be increasingly important in instances such as foodborne disease surveillance and outbreak responses. Thank you for watching this video. If you like this video, please smash the like button and subscribe to this channel for future updates.